Hello students, welcome to EPG Pathshala. I am Dr. Snelata Panwar from the School of Life Sciences, Jawaharlal Nehru University, New Delhi. Today we are going to talk on the module titled Working with Lipids, where we are going to discuss different methods that are used to extract lipids and analyze them from the paper Biomolecules and their interactions. So the main objectives or the aim of this module is to introduce you all to the different methods that are used for analyzing lipids. And this module will involve the discussion of methods that are basically used in the extraction of lipids and to look into different types of adsorption chromatography methods that are used to separate lipids. And then we will look at methods that are used to analyze lipids subsequently. So there are three different steps that are normally followed when you are working with lipids. So the first step when you need to analyze lipid from a given cell or a tissue of an organism is to extract the lipids from the cell or the tissue. So what are you aiming at when you're extracting these lipids? You're actually aiming at a solution that would be enriched in lipids. And how do you do that? We know that lipids are immiscible in water and miscible in organic solvents. So one of the preferred methods of extracting lipids would be to use organic solvents, homogenize your tissue in the organic solvent, and what you will end up getting is lipids in these organic solvents. So once you have a solution that is now enriched in lipids, the next step that you would like to do is separate these lipids. So there are different kinds of chromatographic methods that are used for separation of lipids. Adsorption chromatography methods such as high performance liquid chromatography HPLC and thin layer chromatography methods are used for separation of lipids. Gas liquid chromatography, which is a kind of partition chromatography, is also used for separation of lipids. Once the lipids have been separated by these various chromatographic methods, the last step that you would need to do is to see the kind of fatty acids that are present in the extract. So you would like to analyze the fatty acid composition, and this is normally done by subjecting the lipids to mass spectrometry. So usually the gas liquid chromatography is attached or coupled to a mass spectrometer. So while the lipids are getting separated from gas liquid chromatography, they enter into the mass spectrometer where they are analyzed and identified by their unique fragmentation pattern. There are also specific hydrolysis methods that can be used to analyze fatty acid composition, and we will go through all these methods in details as we move on in this lecture. So the first step that you have to in order to analyze lipid is to extract the lipids. And as we've already seen, that lipids are immiscible with water. Hence, their extraction requires organic solvents, and the technique is referred to as solvent extraction method. So, solvent extraction method is the most commonly used method for extraction of lipids from both biological or food samples. Once the lipid is extracted from a cell or a tissue of an organism or from food samples, the weight of the lipid is measured by either weighing the remaining sample after the extraction with organic solvents or weighing the extracted lipid sample after evaporating the organic solvent. So you can evaporate the organic solvent by flushing a stream of nitrogen gas into the tube. The organic solvent will evaporate, and what you're left with is a thin lipid film sticking to the walls of the tube, and then you can measure the weight of this tube, which will give you the weight of the lipid sample that you have begun analyzing. So different mixtures of organic solvents are used to isolate different types of lipids. For example, if you are interested in isolating or extracting neutral lipids from a mixture, the solvents that you would prefer to use would be ethyl ether, chloroform, or benzene. And why these solvents? These solvents prevent the clustering of lipids, which normally occurs due to hydrophobic interactions between the hydrophobic tails of these lipids. These solvents prevent this lipid clustering and enables the extraction of neutral lipids from a mixture of lipid. If you are interested in isolating membrane lipids, now you have to remember that membrane lipids will also be interacting with the proteins that are intercalated into the plasma membrane. So when you're isolating membrane lipids, you need to ensure that the interaction between the lipid and the protein is also weakened, besides the hydrophobic interaction between lipid to lipid hydrophobic tails. So in this situation, the solvents that you would prefer would be more polar, like ethanol and methanol, because these solvents will prevent hydrophobic or will weaken the hydrophobic interactions between the lipids. They will also weaken the ionic and hydrogen interactions between lipids and membrane proteins. And by doing so, most of the lipid sample would be enriched in membrane lipids. Since the aim is to extract lipid from a cell or a tissue of an organism, you would have a mixture of or different types of lipids in this mixture. So you would have neutral lipids, you would have charged lipids, you will also have membrane lipids. So you have to decide upon organic solvent system in order to maximize the extraction of lipids from the source that you're interested in. So a mixture of chloroform, methanol, and water is the most commonly used organic solvent mixture that is used for extraction of lipids from tissues. And note here that this solvent system has non-polar solvents as well as polar solvents, which will ensure the extraction of all types of lipids from the tissue. So the tissue is taken and it is homogenized in the above solvent mixture. Once homogenization has taken place, a little water is added to the homogenized sample. As soon as you add water to this homogenized sample, you will see that there are two phases. The top phase will consist of methanol and water, while the lower phase will consist of chloroform. So since the top phase is more polar in nature, polar molecules like sugar and proteins would partition into the top phase, while the lower phase is non-polar in nature because it has the organic solvent chloroform. Lipids will partition into the lower phase. So this is how you ensure the extraction of lipid from tissues. So let's have a look at the two different kinds of solvent systems that are used for extraction of lipids. The first solvent system comprises of carbon tetrachloride, methanol, and water. And like we have discussed before, you see how it's a mixture of both non-polar as well as polar solvents. This particular solvent system is useful in the analysis of plant lipids for separating non-polar carotenes and chlorophylls from more polar phospholipids and glycolipids. So the use of this solvent system has been standardized for the analysis of plant lipids. Another solvent system that you see here is the petroleum ether ethanol water system, which is used to remove polar contaminants when the interest lies in the subsequent analysis of neutral glycerides, which may be then recovered from the ether phase. So the point to be noted here, what you really have to note here is that the solvent system comprises of both non-polar as well as polar organic solvents in order to ensure that both nonpolar as well as polar lipids come into the solvent system, and so your system or your solution is enriched in all kinds of lipids. Once you have a sample that is enriched in lipids, it's the crude lipid extract that you have. The next step that you would like to do now is to separate these lipids, individual lipids, from this lipid mixture. So in order to do that, we perform adsorption. You make use of adsorption chromatographic methods. So what is adsorption chromatography? In this method, separation is based on polarities of the lipids in a lipid mixture. High performance liquid chromatography, HPLC, and thin layer chromatography, TLC, are two different kinds of adsorption chromatographic methods that are used to separate lipids. In both the types of chromatographic methods, the adsorbent that is used is silica gel, which is polar. Silica is a form of silicic acid, and since it is polar in nature, what do you expect? You would expect that the polar lipids in the lipid mixture will bind to this adsorbent. So silica gel is the stationary phase in this chromatographic method. So what is the mobile phase here? In HPLC, the glass column is packed with silica gel, while in TLC, a thin layer of silica gel is spread onto a glass plate. In both the cases, the lipids will adhere to the adsorbent based on their polarity. In TLC, the silica gel is spread as a thin layer onto a glass plate. In both the cases, the lipids will stick or bind to the adsorbent based on their polarity. So like I said, more polar lipids will bind tightly to the silica gel and will be retained for a longer time in the stationary phase. So in adsorption chromatography, the stationary phase comprises of silica gel, while the mobile phase will comprise of a mixture of organic solvents. So the lipids that are polar would bind to the fine particles of the adsorbent by polar, ionic, and van der Waals forces, and therefore they will be retained for a longer time in the column or on the TLC plate. The most polar molecule will be held most tightly to the adsorbent, and the separation would take place according to the relative polarities of the individual lipids. So, how would these lipids be eluted out if they are tightly bound to the adsorbent? So, there is a mobile phase that is used in adsorption chromatography. And in the case of HPLC or TLC, in the case of HPLC, basically, the mobile phase will consist of a solvent system that will be used in gradients of increasing polarity. Solvents of increasing polarity are used, where the least polar lipid will emerge in the early fraction, while the most polar lipid will emerge in the later fractions. So the table that you see here lists compounds according to their polarity. So you see that saturated hydrocarbons are least polar, while lysolecithin is most polar. So if you have to separate lysolecithin from saturated hydrocarbon in adsorption chromatography, and if you're using solvents of increasing polarity to elute these two compounds, the least polar lipid will emerge in the early fraction because you're using a gradient solvent system with increasing polarity. The least polar lipid is saturated hydrocarbon, so it will emerge first, while the most polar lipid is lysolecithin, so it will emerge later in the fractions. There are different kinds of solvents that can be used for eluting different types of lipids. So here you see that if you have to separate a mixture of neutral lipids, the column used would have silicic acid as the stationary phase, and the solvent that you would prefer to use would be hexane, which is nonpolar, containing increasing proportions of diethyl ether from a range between a range of 0 to 100%. So you can increase the proportion of diethyl ether in hexane in order to separate a mixture of neutral lipids. And the box on the right it shows the order in which the neutral lipids will be eluted. If you're interested in separating a mixture of glycolipids and sulfolipids, then the preferred solvent system would be chloroform and acetone in a 1:1 ratio. If you have to separate a mixture of phospholipids, the preferred solvent would be chloroform and methanol in a ratio of 95:5. If you keep increasing the proportion of methanol to 50-50, it would result in the elution of phospholipids in the order as shown on the right side. So we saw that the general principle in adsorption chromatography is that there is a stationary phase, a polar stationary phase, and there is a carrier phase. The stationary phase is polar, so polar lipids will bind to the stationary phase tightly, and the carrier phase can be a mixture of organic solvents depending on what kind of lipids you're interested in looking at. You will use different solvent systems in order to elute the bound lipids from the column. So in case of high performance liquid chromatography, the column is packed with silica gel, and the column has a very small diameter. So since this column has a very small diameter, the solvents are passed through the column under high pressure. So while the solvents are passing the column along with the mobile phase, the polar lipids will bind to the silica in the column, as silica is polar, and the neutral lipids, because they are uncharged, they will pass through the column in the first wash with chloroform. So the first eluate will contain a mixture of neutral lipids. Then the polar lipids will be eluted by washing the column with solvents of increasing polarity. So the order of elution would be that if you are using acetone as the solvent system to elute the lipids from the HPLC column, uncharged polar lipids will be eluted first. And if you're using methanol, very polar or charged lipids will be eluted first. Thin layer chromatography is another kind of adsorption chromatography, and like I said, it follows the same principle of separation as as we saw for HPLC. The only difference here is that the silica gel, instead of being packed into a glass column, is actually layered onto glass plates. The function of TLC is to separate lipids on either a preparative scale or on an analytical scale. So sometimes TLC is also used to fractionate the lipids into classes prior to removal from the plate and further analysis by gas liquid chromatography. So the principle of separation remains the same. Since the silica gel is layered onto glass plate, the stationary phase is polar. So the most polar lipid will be held most strongly to the adsorbent, which is silica. The mobility of the lipids during TLC will be affected by, again, the polarity of the solvent. Increasingly, polar solvents will break the adsorptive bonds with greater efficiency, resulting in greater RF values. RF is the retardation factor, and we will see what it stands for and how it is calculated as we move on. In thin layer chromatographic plates, silica gel is coated onto a glass plate or metal foil plate, and this comprises the stationary phase in TLC. So you can either prepare silica coated glass plates in the laboratory, or you can buy pre-made TLC plates from companies where they are commercially available. So once you have this plate, the test sample is applied as a spot or a thin band at one end of the plate, and we will see how that is done as we move on. The plate is then placed in a glass chamber that will contain a mixture of solvents. The solvent will move up the plate by simple capillary action, and as the solvent will move, it will transfer analytes in the sample at a rate that will be determined by the distribution coefficients of the analytes between the mobile and the stationary phase. The analyte movement will stop once the solvent reaches the end of the plate. So by distribution coefficient, we mean that since the more polar solvents will adhere to the polar adsorbent silica stationary phase, the non-polar ones will bind with less affinity to the adsorbent phase. The non-polar ones will move faster if the solvent system is non-polar, while the polar molecules will move slowly on the TLC plate. So here you see the different steps that are done in order to start running a TLC plate. So first step would be the preparation of plates, as I've already discussed. You can either make your own plates or you can have pre-prepared plates bought from a company. And in both the cases, a slurry made of silica and water is spread onto these plates. So silica is the adsorbent and the stationary phase in any of those TLC plates. The sample is dissolved in organic solvent and applied on one edge of the plate using a micropipette. About 12 to 15 samples can be applied as discrete spots on one TLC plate. This plate is then placed in a glass chamber which will contain the mobile phase. So once you place the plate in the mobile phase, you expect that the mobile phase, which consists of an organic solvent system, will start moving up the plate by capillary action. Before you place the plate in the glass chamber, you fill the glass chamber with the organic solvent system and allow the tank to equilibrate for about 1 hour. After this equilibration has been attained, the plate will then be placed vertically in the tank with the edge near the origin in the solvent system. The separation is allowed for about 20 to 30 minutes for analytical procedures and for about 75 to 90 minutes for preparative procedures. Once the solvent has reached the other end of the plate is removed, and then there are different methods by which the spots on the plate can be detected. Stop. So once the separation of the lipids has taken place onto the TLC plate and you remove the plate from the glass chamber, you can detect the analytes in the ways that are listed here. There are different ways by which you can do it. You can spray the plate with a specific reagent that will convert the analyte into a colored product that is visible to the naked eye. So normally what you do is you will take the plate and place it in a chamber that has iodine vapors. Iodine will bind to the lipids reversibly and color them, and you can see the spots colored with naked eye. The second way by which you can detect the analytes on the plate is by viewing the plate under ultraviolet light, assuming that the analytes will absorb in this region. Another way of detecting analytes on the plate is by incorporating a fluorescent dye into the stationary phase, so that when the plate is viewed under ultraviolet light, the analytes show as blue, green, or black spots, as you can see in the figure depicted shown here. So this setup is a typical setup that is used for running TLC plates. The colored spots here correspond to individual lipids. The arrow that is marked as the origin is the is one edge of the plate where the lipid samples were initially spotted as spots. So these samples, as I told you, were dissolved in organic solvents and then spotted onto one edge of the plate. The glass chamber contains this a system of organic solvent. The one in the sky blue depicts the solvent system. So once the plate is placed in this glass chamber, the solvent system will start moving up the TLC plate due to capillary action. You can then measure the retardation factor, which is basically a method by which you can calculate the movement of a given analyte on a TLC plate. So how do you calculate this retardation factor? Retardation factor is basically the distance moved by the analyte from the origin. It's called dA, over the distance moved by the mobile phase front from the origin, which is dF. As you can see in the figure, the pink arrows here depict the distance that has been moved by the analyte, and the distance that the organic solvent has moved onto the plate. So by measuring these distances, you can easily calculate the retardation factor of these spots, compare it with a reference or a control that you have run on the TLC plate, and then you can identify the individual lipids when you against a reference that are using on the TLC plate. There are two types of gas chromatographic methods that are used: gas solid and gas liquid. Gas solid chromatography is an automated type of chromatography in which the mobile phase is an inert gas, while the stationary phase is a solid, and hence the name gas solid. The separation in this chromatographic method is based on the molecular size and shape, wherein the smaller molecules can be temporarily retained in the stationary phase. The gas-liquid chromatography, on the other hand, is a type of chromatography where the adsorbent that is used, or the stationary phase, is a liquid, and hence the name gas-liquid. So basically, in gas-liquid chromatography, the mobile phase, just like gas-solid chromatography, is also an inert gas. Here, the substances to be separated are moved by an inert gas along a tube that is filled with finely divided inert solid coated with a non-volatile oil. So basically, the stationary phase here is a non-volatile oil that is coated onto a column. The migration of each component in gas-liquid chromatography is determined by its solubility in oil and its vapor pressure. So gas-liquid chromatography is a very useful and powerful technique that is used in analyzing complex mixtures of lipids at a very high resolution. The advantage of using GLC for analyzing lipids is that you can qualitatively as well as quantitatively analyze a large number of compounds that have low polarity in the lipid sample. GLC is generally coupled to mass spectrometry, making it a very powerful analytical technique because the samples that come out of GLC are directly subjected to mass spectrometry, where they are analyzed based on their unique fragmentation pattern. Another kind of chromatographic method that is used to separate a mixture of lipids is similar to HPLC and is called the gas-liquid chromatography, but the apparatus is very different from that of HPLC apparatus. The principle of separation in GLC is that the compounds are separated based on their volatility. Unlike HPLC or TLC, the GLC consists of a stationary phase which is liquid in nature. So what is this liquid phase? This liquid phase consists of a high boiling point liquid material like silicon grease or wax that is coated on the inner side of the column. The mobile phase here is a gas, which is an inert gas like helium and nitrogen, which is also different from HPLC and TLC, where the mobile phases are a mixture of organic solvent, while in GLC, the mobile phase is an inert gas, and hence the compounds need to be volatile in order to be carried by these carrier gas through the column. The separation is based upon the partition coefficients of the volatilized analytes, which means that the time that the volatilized analytes will spend between the stationary and the mobile phase will decide which compound will be carried faster by the carrier gas. Therefore, GLC is mostly used for those compounds that are volatile but are thermally stable as the temperature of the column is raised to 50 to 300 degrees Celsius to facilitate the volatilization of the analyte. Because the temperature is increased to such high degrees, the compound needs to be thermally stable, as I mentioned here. So lipids that are naturally volatile will become volatile when the temperature of the column is increased to about 300 degrees Celsius and will be carried by the inert gas out of the column, while lipids that are not naturally volatile then need to be derivatized first by there are different derivatization process. One such process is by transesterification. So you derivatize the lipids prior to injecting them into the column to increase their volatility. Here you see a list of the components that make up a GLC system. So a gas liquid chromatography system comprises of a column that is housed in an oven that can be temperature programmed, and this column is basically houses the stationary phase, which is, like I said, a high boiling point liquid. Then there is a sample inlet point. There is a carrier gas supply attached to the system, which is basically the mobile phase, which will carry the lipid mixture along with it through the column. And then this GLC system is attached to a detector, amplifier, and a data recorder system. So you see a figure here. This figure shows the different components of a typical GLC system. So the box here where you see this capillary column written is basically the oven, which is temperature programmed. The capillary column here is a long coil. This coil is not much thicker than a human hair, and it's about 30 meter in length. It's a non-polar column. And it's the inside of this column, the walls of this column are filled with the high boiling point liquid like silicon, grease, or wax, which is basically the stationary phase in the GLC system. The purple container here, which is marked as the carrier gas container, is the container that carries the inert gas like helium. So this inert gas is blown through the tube, and just at the top of the column where you see the arrow showing the injection port is there is a preheater. So what is the role of this preheater? The syringe here on the top of the injection port is a micro syringe which can hold up to 1 microliter of sample. And this micro syringe is used to inject the sample into the column. The preheater that is present near the injection port will immediately vaporize or make the sample volatile so that it can easily be carried by the carrier gas through the column. So initially, everything in the column will move together, okay? But then based on the affinities of the analyte towards the stationary phase and towards the mobile carrier gas phase, the mixture, the lipid mixture would start to separate into individual lipids. So as the lipids will continue, as the lipid mixture or the individual lipids that are getting separated will continue to move through the column, they will then eventually, once they reach the end of the column, they will pass through a, they will reach the end of the column, and then they can either go through a flame which is used as a detector, and that is called a flame ionization detector, or they can go through a box that you see here, which is a mass spectrometer for further analysis. So the mass, these days, GLC systems are coupled to mass spectrometer, which is the universal detector in these systems. So as the eluent will come out at the end of the GC column, they will go through the mass spectrometer. In the mass spectrometer, the eluents will go into the electron gun through a series of magnets, and these magnetic field will smash the molecule into small pieces, and these pieces are fed between some electromagnets which are very powerful, which these magnets will then deflect these small pieces according to their mass. And this detector, which is the mass spectrometer here, is then attached to an integrator, which is basically a computer. So once this deflection is then recorded by a plate, which consists of many electrical circuits, as a result, we get a mass spectrum, which is also called a mass chromatogram. And the peaks that you see here are basically peaks that represent individual lipids in the mixture. These peaks that you obtained out of a mass spectrometer are then compared against a database which has about 200,000 to 300,000 molecules listed in it. And then according to after the comparison, you will find out which peak corresponds to what molecule. The columns that are used in GLC are of two types. You can either use a packed conventional column, which means that the columns are packed with stationary phase coated onto an inert silica support. Or you can use open tubular columns, which are made up of high quality fused quartz and are about 10 to 100 meter long and 0.1 to 1 millimeter internal diameter. So let's just go through the list of steps that I mentioned in the previous part that are used to inject the sample into the GLC column. So the less polar or the non-polar compounds can directly be injected into the column. The polar compounds will be retained on the column for a very long time if they are applied directly. So like I said, the polar compounds therefore will need to be derivatized before they are injected into the column. And in order to do so, the polar compounds can be derivatized by selenization, methylation, or perfluoroacylation. These are different methods by which polar compounds can be derivatized, and these derivatized compounds then become more volatile. The sample is then dissolved in solvents like acetone, heptane, or methanol and injected using a microsyringe, as I mentioned earlier. And this injection is usually automated. And as I mentioned earlier, the injection region has a preheater which keeps the injection port at a slightly higher temperature than the column itself, which ensures a quick and complete volatilization of the sample as soon as it is injected into the column, so that it can be carried by the carrier gas immediately as it is injected. So the carrier gas or the mobile phase, like we've already seen, is an inert gas like helium or nitrogen. So this inert gas, once before it's flushed into the column, it is pre-purified to remove oxygen, hydrocarbons, or water vapor. And the flow of the carrier gas is also maintained by a controller. The flow needs to be regulated because that will also decide the retention time of a particular analyte. So As we saw previously, the GLC system can be connected to different kinds of detectors, and one kind of detector that I mentioned was the flame ionization detector. There's a nitrogen phosphorus detector that the GLC system can be coupled to, but like I said, mass spectrometer detector is one of the universal detectors that is used to detect analytes once they come out of the GLC column. So let's have a look at the applications of gas liquid chromatography. So as we've already seen, GLC is confined to volatile and nonpolar compound separation. The analytes are characterized by their retention times relative to a standard reference compound or a control. The most volatile compounds are carried by the carrier or the mobile gas, which is the inert gas. And since they're most volatile, they'll be carried by the mobile phase. They will elute first from the column, while the less volatile compounds will get dissolved in the column material, which is the stationary phase, and hence will elute later. So the time that they spend in the stationary phase will be more, or in other words, you can say that they are retained in the stationary phase for a longer time. So using this technique, mixtures of fatty acids with various degrees of unsaturation and different chain lengths can be resolved. Coupled to mass spectrometry, this is a very powerful technique which leads to the identification of individual fatty acids with different degrees of unsaturation and different chain lengths. So as the eluates come out of the GLC column and enter the mass spectrometer detector, mass spectrometry analysis will establish the hydrocarbon chain length and the position of the double bond in the fatty acids. Using a mass spectrometry analysis, similar lipids such as two fatty acids that have the same chain length but are unsaturated at different positions will elute together from the column, but when they are sampled by mass spectrometry based on their unique fragmentation patterns, they can be separated or identified simultaneously. So what I mean to say here is that if there are two fatty acids that have the same chain length and also the same degree of unsaturation, but they only differ in the positions of the double bond in their chain, they will also be, you will be able to identify them by their unique fragmentation pattern that would be generated when they pass through the mass spectrometer. Here you see these schematics depicting a gas chromatography mass spectrometry analysis. So in A, you see that this is a column which is a part of the GLC system. So you inject a sample here. The analytes that have a high affinity to the stationary phase will move slow in the column because they'll be bound to the bound to the stationary phase here. The arrow on the top shows the direction of the flow of the inert gas, which is the mobile phase, and it's helium here. You see that these red analytes, since they have low affinity to the stationary phase, they are carried by the inert gas faster, and so they will elute out of the column first and would be fed into the detector, which is a mass spectrometer in this case. Once they are fed into the detector and analyzed by the mass spectrometer, this is the kind of chromatogram that you will see as is shown in part B of this figure. The peaks here represent individual analytes, and this is the x-axis is the time, while the y-axis depicts the relative abundance of the lipid. The area under each peak will tell you the mole percent of the analyte that was present in the mixture. So you can calculate the retention time or the relative retention time, which is defined as the time between sample injection and an analyte peak reaching a detector at the end of the column. Okay, so in this situation, the red analytes which will reach the column faster, so they will appear faster. So the first peak that you will see in the chromatogram would be of these analytes, followed by the analytes represented in blue, which, since they have high affinity to the stationary phase, they will elute later. And so they would have spent a lot of time or would have been retained in the column for a longer time, so they will have a higher retention time. So all the techniques that we have discussed so far are used to analyze individual lipids or separate individual lipids from a lipid mixture. And all these techniques can be made use of in order to look at the complete lipid profile within a cell or a tissue or an organism. And if you are performing this kind of a study where you're looking at the complete lipid profile of a cell, you call this as lipidomics. So how do you define lipidomics? Lipidomics is a large-scale study of pathways and networks of cellular lipids in biological systems. And the data that you generate when you're doing a large scale study of pathways of cellular lipids is called a lipidome, which is basically the complete lipid profile within a cell. So what do you do in lipidomic research? So lipidomic research involves analyzing which lipids are present and in what proportions in a given situation in a cell. It involves analyzing interactions of lipids with other molecules. It also involves quantitatively describing the alterations in lipid content and their composition in response to a perturbation of a cell. Perturbation could be a stress condition which could have triggered alterations in lipid content, and you can monitor the alterations in lipid content and their composition by performing a lipidomic study. So how do these studies help? These studies help in defining the biochemical mechanisms of lipid-related disease processes through identifying alterations in cellular lipid metabolism, trafficking, and homeostasis. So you can compare the lipid profile of a normal cell with a diseased cell and look into the alterations that would have occurred in the diseased cell in terms of the lipid content and in terms of the lipid composition. Here you see a flow chart that depicts the final outcome that you would get if you're performing a lipidomics study. So you can apply all the techniques that we have studied so far, like GLC, HPLC, separate the lipids, analyze them through mass spectrometric techniques, plus high throughput, plus high resolution would enable you to receive or obtain a quantitative catalog of all the lipids that are present in a specific cell type under a particular condition. And it would always also enable you to look into the changes that have occurred in the lipidome in diseased condition or during differentiation, etc. So the gist is that by applying all the techniques, high throughput techniques and high resolution, you would be able to get the complete lipid profile of a given cell, a normal or a disease cell, and then you can compare the lipid profiles of normal versus disease cell and see what are the lipids that are altered in a diseased condition. So students, let's now summarize what we have learned in this module. So we have looked at the different steps that are used for analysis of lipids in a lipid mixture. The first step as we have discussed in determining lipid composition from tissues, is extraction of lipids with organic solvents. Once you have a fraction that is enriched in lipids, these extracted lipids are then separated by different kinds of chromatographic methods like thin layer chromatography, gas liquid, or high performance liquid chromatography techniques. Individual lipids that are separated now by using these different chromatographic techniques can be identified by either their behavior on the chromatography columns or by mass spectrometry. In the end, we also saw how lipidomics is an extremely strong technique that is used to determine the lipid profile in a cell or a tissue of an organism in response to different stress conditions or during differentiation. Thank you.